Hello everyone, we are a part of the 2024 C-Phages cohort at La Sierra University, and we would like to share with you our research on the H&H endonucleases in phage bugger from cluster GB.
H&H endonucleases have a variety of functions in phages. They contain a NUMOD4 DNA binding motif, which is involved in binding and recognizing specific sites of DNA, and an H&H motif, which cleaves DNA sequences. During our bioinformatic analysis of bugger's genome, it was found that bugger has 4 H&H endonucleases. This is significant because the other members of cluster GB only contain 0 to 2 H&H endonucleases in their genome. These findings inspired further analysis to understand why phage bugger contains more H&H endonucleases than its cluster mates, and the role that these additional proteins may have in bugger's life cycle. The approach we took for this project began with comparing the amino acid sequences of all the H&H endonucleases found in cluster GB phages using UniProt. We then arranged a percent identity table that displays the likeness between all H&H endonucleases. Comparison between the 4 H&H endonucleases in bugger were then carried out using Chimera X in order to identify structural differences that might be present despite their conserved domains. In addition to a guided tree to show the relationship between H&H endonucleases from cluster GB.
Our gene alignment comparison shows the sequence similarity between the 7 H&H endonucleases present in cluster GB phages. These include 4 from phage bugger, 2 from phage Leroy Jenkins, and 1 from phage Casita. The dark purple color highlights entirely conserved regions between all of the sequences. The medium and lighter purple shades identify regions that are partially conserved. The alignment also shows that bugger H&H endonucleases differ in sequence and in length. The gene percent identity matrix in Table 1 displays a numerical representation of the sequence homology between the H&H endonucleases. The data shows that bugger gene 84 is 99.41% similar to Casita 88 and 100% similar to Leroy Jenkins 94. Additionally, bugger gene 103 shows a 99.35% similarity to Leroy Jenkins gene 114. The high alignment percentages between the H&H endonucleases in bugger, Leroy Jenkins, and Casita suggest that they have similar structures and functions. The table also shows that the 4 H&H endonucleases from bugger have lower than 40% sequence similarity, strengthening the idea that each of them have different functions and target sites.
We use the amino acid sequences to generate 3D predictions of the protein structures using AlphaFold. The structural comparison of bugger's H&H endonucleases emphasizes the variations that would allow them to interact with different target sites. Both the left and right sides of each structure contain similar traits, but upon close inspection, distinct regions within each of the figures can be identified. In the AlphaFold coloring dialog, the confidence levels in the accuracy of the predictions can be interpreted as follows. High confidence in the residues are colored in blue, medium confidence in the residues in yellow, and low confidence in the residues will be depicted in red. Additionally, a guided tree was generated using UniProt to display the relationship between the H&H endonucleases present in cluster GB. The length of the line dictates the closeness between each H&H gene. Furthermore, even though bugger genes 84, 86, and 103 were placed in the same font, the guide tree shows that bugger 86 diverges from genes 84 and 103 and is more closely related to gene 62. Bugger 84 is identical to Leroy Jenkins 94, and both are closely related to Casita 88, but all three differ from bugger genes 62, 86, and 103. Bugger 103 is more closely related to Leroy Jenkins 114 than to the other endonucleases in phage bugger. Finally, bugger genes 62 and 86 are less closely related to the other endonucleases in cluster GB, with 62 being the most different from the rest. In summary, our analysis suggests that the endonucleases in phage bugger are different from each other, and as a result, they likely target different sequences and perform different functions. Further research can allow us to identify the targets of the H&H endonucleases, leading us to discover each of the possible functions of these proteins and their relevance in phage bugger's life cycle.
Thank you!